This video is for Chapter 2. We're going to start off looking at Section 1, which, actually before we get to Section 1, we're going to look at the core case study. So make sure that you go through this and look at how this is an example of a controlled experiment, what is a control group, what are variables, and of course, what is the experimental group. Over on the first page of Section 1 is a review of the scientific method process. It's important to remember that a lot of times when we do labs in a science classroom, we generally try to follow this method, but understand that a real scientist or somebody who's doing research isn't necessarily going to follow it in these specific steps every single time. But this is an important process. So this particular model right here is a flow chart that was based off of what happened with this experiment over here. So, but it uses all of the same things. Like, for example, before you can do any experiment, you have to identify what is the problem, or what's the problem that we're trying to solve, or what's the question that we're trying to answer, which would be down here. So it depends on which step we're at. We're applying knowledge that we already know. We're performing an experiment. We're analyzing data. We're proposing hypotheses to explain that data. And then, of course, there's always revisions. There's always peer review, which means making sure that other scientists can look at our work, can either replicate our work or refute our work, meaning there's something that they're saying that we did wrong, or they come up with contradictory ideas. So there's a whole process to it. So make sure that you know this figure and caption. And by the way, when I say caption, it's this description down here. So diagrams and then the paragraph underneath are always really important with textbooks, but students usually tend to skip over them. All right. Make sure that you know these 4 steps, or these 4 things about why critical thinking is important to science. Make sure that you understand what is the difference between a hypothesis, which is our or a researcher's potential answer to their question, versus a theory versus a law. So why is a law going to be much more conclusive and much more kind of, not evidence, but I can't think of the right word I'm trying to say, but why is a law so much higher or ranked so much higher? So what's the difference between these 3? All right, moving to the next page, looking at the science in focus. This is looking at the case study of Easter Island, which Easter Island is a small island where there was a collapse of a civilization, and for a really long time it was believed that the civilization collapsed because of environmental degradation. The Polynesians that lived on the island, basically they used their resources faster than they could reproduce. They cut down all their trees, and eventually their civilization collapsed because of lack of resources. So that was the original hypothesis. Around 2006, there was an anthropologist who came along who kind of reevaluated that hypothesis and looked at different pieces of evidence, and you can go back and read that on your own. But basically, he developed a new hypothesis to explain why the civilization collapsed, which was saying that yes, even though the people were using their resources unsustainably, in fact, it was actually rats that were brought with onboard ships as stowaways that basically were responsible for the collapse of the civilization, because they rapidly multiplied and they ate all the tree seeds and all the plant seeds before these plants could regrow. And so the idea behind this is that a new hypothesis is always possible based on a revision of data. So this first initial hypothesis was the commonly held belief for a long time, but again, with new data, it was revised and changed to a different form. All right, make sure that you understand and you're clear on what do we define as reliable science. What are the differences between reliable and unreliable science? So here are these statements here. These are things that help us figure out or help us know why is this particular science reliable compared to others. Okay. Also, understanding that science has limitations. So make sure that you go over and you're aware of what are the there are 4 different limitations of science. Science, there's going to be limits to what it can do. Okay. Moving on to section 2. Section 2 is looking at a review of chemistry. So if you've had chemistry, this should be super easy. If you had bio, it may or may not have been covered in the beginning. So if you either don't remember or if you are having trouble understanding section 2, please make sure you make time to come and see me. But for this beginning first section, understand what is matter. What are the 3 physical states of matter? So that's where we have solids, liquids, and gases. What are the 2 chemical forms of matter? And then moving over to the next page, when we start talking about atoms, make sure you know atoms, make sure you know atomic structure. So what are they made of? They're made of protons, electrons, and neutrons. Protons and neutrons are in the center called the nucleus. Electrons are orbiting around the outside in this potential, and it's not going in a circle. It's 3-dimensional, which of course never shows in a picture, but that's called an electron orbital. And know that protons are positive and electrons are negative. Make sure you understand that what an ion is. So an ion, which is actually talked about over here, is a particle with an excess positive or negative charge, a molecule with an excess positive or negative charge. Make sure you know what isotopes are, because we will definitely go back and talk about those a lot when we talk about nuclear energy and half-lives and all of that fun stuff. This table up here, make sure you recognize these symbols for these positive and negative ions. Is it necessary for you to memorize them in this particular chapter? Not necessarily. I would definitely say make sure you know hydrogen ion, sodium ion, hydroxide ion, but these are things that we're going to see throughout the course, especially when we talk about biogeochemical cycles and different types of pollution. All right, going to the next page. Here, and your book does not have a diagram of a pH scale, so make sure that you're aware, or make sure that you remember what a pH scale looks like. So what does pH measure? What are the characteristics of an acidic solution? What are the characteristics of a basic solution? And when I say characteristics, meaning what's the concentration of what type of ions? Okay, and then remember, what is a pH scale? What's the range for neutral, which is 7? What's the range for acids, which is anything less than 7, so 0 to 6 roughly. And then strong acids, of course, are going to be closer to 0. Bases are going to be above 7, so 8 to 14, and strong bases are going to be close to 14. Remember, and this is not in the book, I'm just putting this here as a side note, remember that in the pH scale, each number, so the 1 through 14, equals 10 times the hydrogen ion concentration. So if you're going from a pH of, say, 7 to all the way up to a pH of 8, that's not a jump of 1, that's a jump of 10. And then if you're going to, say, a pH of 7 up to a pH of 9, that's not 20, that's 10 times 10. So that's actually an increase of 100 times the hydrogen ion concentration. Remember down here when they talk about organic compounds versus inorganic compounds, organic compounds are carbon-based. Really, we're going to talk a lot about these later on in the year, especially when we talk about energy and pollution. We'll go back and see hydrocarbons and chlorinated hydrocarbons. These up here, different compounds that are used in the textbooks, start getting familiar with those again, because they are going to come back and you will see a lot of these again. All right, next page. So just kind of know this figure and caption. This is a review from bio. Remember what genes are, remember what traits are, or characteristics. Chromosomes, which are the bundles that DNA is put into as cells are getting ready to go through cell division. All right, and then make sure you're clear on what's the difference between a physical change versus a chemical change. And then the last part of section 2 is understanding what are the 3 types of nuclear change. So make sure you're familiar with radioactive decay, nuclear fission with an I, and nuclear fusion with a U. So what are they? Those are the 3. when does each one occur, which tells you right here under this post-it. So make sure you know this diagram and this caption. And then the last part, understand what is the law of conservation of matter saying. So it's saying that as things are going through chemical reactions, as things are happening, matter cannot be created from nothing, and it cannot be destroyed either. So whenever you have a chemical reaction occurring, it's basically just a rearrangement of those atoms from one form to the next. So it's like saying you have Legos, and you're taking two groups of Legos and you're pulling things apart and you're rearranging them to make either one big thing of Legos or one big product, or you're making two differently shaped products. Everything is still there, it's just a different rearrangement.